Hello, everybody. Good afternoon. I hope you enjoyed your lunch and had a little bit of a break, got a chance to connect with people. My name is Shayunika Mohanta. I am the Strategic Marketing Manager for Epigenetics Applications, and I am thrilled to welcome you to today's workflow demo for Epitranscriptomics with direct RNA sequencing. So, of course, unlike other traditional RNA-seq methods, which rely on things like reverse transcription, amplification, and can therefore have certain biases, our approach, of course, uses native RNA molecules directly, which reveals features that are often lost or distorted in cDNA-based methods. With direct RNA sequencing, we can detect key RNA modifications. If you came to the the tech update yesterday, you saw all the new modifications that we've now been able to do. But some of the key ones, of course, include M6A, inosine, M5C, etc. We can also capture full-length isoforms without any fragmentation, thus preserving the biological context and complexity. We can also estimate the poly-A tails very accurately, which is crucial for understanding RNA stability as well as translation. So this workflow is, of course, available in real time through Minnow as well as Dorado, therefore empowering you to see the full complexity of the transcriptome as well as the epitranscriptome in a single run. So with that, we're going to move on to our Q&A session. And I am delighted to be joined by Anna Wieczajko. Some of you may have heard her amazing breakout session talk yesterday. But as a brief reminder, Anna is a bioinformatician in diagnostics at the Institute of Human Genetics, University Medical Center, Mainz. Throughout her research, Anna's primary focus has revolved around the analysis of next-generation and third-generation sequencing data, investigating transcriptomic factors within mouse models and clinical contexts. She does direct RNA sequencing and, of course, detects RNA modifications. And as an added bonus, we're also going to be joined by Anna's lab mates, Stefan and Johannes. So please welcome them to the stage. Thank you so much, guys, for joining us on this Q&A session. We're going to kind of switch back and forth, taking questions from the audience, as well as some that I have for you as well. So we'll take the first question from the audience. If you do have one, if you could please raise your hand, and we can make sure to bring a microphone to you. All right. Anybody brave enough to go first? Yes, the gentleman here. Thank you.
Hey, guys. So the library preparation protocol for direct RNA sequencing is based on making a cDNA backbone, and that is attachment from the poly-A tail of mRNAs, and then the cDNA backbone is written. But the protocol also says that you can do mRNA enrichment or RNA depletion. Which is the best way to do it? Just the whole RNA and sequence it as it comes, or RNA depletion or mRNA enrichment? Is there any benefit of doing mRNA enrichment if the library is created from poly-A tailed RNA anyway?
Johannes, would you like to take that?
I would like to.
Or Stefan.
To Stefan, because
so
lab background.
yeah, I'm actually working a lot with ribosomal RNA, so I'm not trying to deplete it. But if you use the normal protocol with the poly-A tailing, with the alignment, annealing to the poly-A's and the ligation, the proportion of mRNA is quite high already, so I never tried to enrich. I was already quite happy with what I got. But as I said, I'm usually doing in vitro polyadenylation to capture ribosomal RNA, so it's hard to comment. But you can really play with these kind of styles to enrich for certain populations for sure. And for us, it's usually working out if we're also playing a bit with the techniques. So yeah, whenever you want to do RNA depletion, I would assume it would work well and you will get an enrichment, but it's just an assumption. Yeah.
Thank you so much. So a follow-up to that, can you guys address some of the challenges that the direct RNA sequencing allows you to address that maybe other technologies cannot?
So first of all, to assess RNA modifications, of course.
Of course.
Which is not possible. So besides all the other levels that can be detected, including gene expression, splice isoforms, and so on, in just one technique, which is not possible with any other technique. I think this is the highest benefit of using direct RNA sequencing, and it is easy to implement into a routine workflow if you have a machine in your lab. So
Yeah.
I think these are the main benefits of using direct RNA sequencing.
Of course. Is there something you guys wanted to add?
No.
Okay. RNA modifications, heard it here first. So now that all three of you are here, did you guys have any specialist training to get up and running, or was it pretty straightforward to integrate the direct RNA workflow into your lab?
So as we discussed already before, we are especially of the opinion that you need an expert in the lab as well as a bioinformatician expert, because we also have our lab wet person here who sequenced all of our direct RNA sequencing. And I think in the beginning, because it was not completely straightforward, but more or less straightforward. But also during experiments, there can be some errors that needs to be discussed. So it's always good to be close together, wet lab and dry lab.
Yeah.
But yeah, maybe you can.
Yeah, but it's straightforward once you have experts, because it's only minor things happening, but
Yeah.
you need to know them. That's the the thing. Yeah.
Great. Is there any other questions from the audience? Yeah. Could somebody give him the microphone?
Hello, I want to ask a question about Dorado. You know, Dorado provides the base calling model and modification model, calling model. And modification model will return the model back. What's the difference of with the model back and the no model back? I want to know the difference about sequence. You know, it can be converted to the FASTQ. What's the difference between the FASTQ converted from the normal bad file and a model bad file?
Okay. So the modification signal is basically stored in the unaligned BAM format usually. So or in the BAM format actually. So you can. I think the FASTQ will not change actually, because it's
so
too.
if we can, if we pay attention to the modification, we can scan the normal base calling and call the M6A directly and use the sequence from the.
Yeah, so I think it's two different models that are operating on that. So the Dorado model is doing the base calling, and then the bases that have been called, basically, so if it's an A, for example, then you're going for the M6A model. Then every spot where you have an A will be actually analyzed with a second model, which is then the M6A model, and it will decide whether it's an M6A modification or not.
Okay, thank you. And another question is, now I noted that there's new four modification model have proposed 2.0 mass, and now we have more than 8 modification calling and modification calling model, and if I want to pay attention to the modification, I will run it again one by one. It's time consuming. Will it speed up in the future? I want to know.
I think
Should
I
I?
think yesterday it was mentioned that you can run it in just using one base calling run. I guess it needs more time, of
Yeah.
course, but I think it wasn't mentioned how much more time it takes to call all of the 8 modifications. I don't know if you have more
I
information
don't think
on
we
this.
have quite an answer since we just released it, but something definitely to explore with our team.
Okay, thank you.
Thank you so much.
Yeah, I would also maybe add something. So I don't know how it's done in ONT, but I would assume that the modifications for certain base identities would be combined. So if you have modifications on an A, for example, then you would call for all the modifications on that A to reduce the amount of models you use. But that's just like something I would do as a deep learner. Like, yeah.
Some great
Yeah.
experimental design. All right. So you guys are, of course, using the Promethean 24 for your research. What aspect of the device, whether that's output, throughput, flexibility, accessibility, what do you guys like, and what really makes it ideal for your lab and the type of research you're doing?
So I think all of them are so we need high throughput because when we use direct RNA sequencing, for example, also for clinical cases, we need high coverage of transcripts. So we need high throughput, flexibility because we have sometimes runs that we need to start side by side or just 5 hours later. And also the accuracy is a bit higher than using min ions. And yeah, I think this is from the clinical perspective, the needs that we
Nice.
need.
Did you two want to add anything to that?
Yeah, I think especially for a clinical context or for us in the research, we are really aiming for getting the best accuracy. This is also a very important point, and that's also why we also call with the submodel. And yeah, that's why the Promethean is perfect for us.
Nice. Is there something you wanted to say? Okay. Any other questions from the audience? Okay, no worries. So when you guys are analyzing poly A tail lengths or even isoforms, how do you generally ensure quality and reliability in your experiments?
So in our recent manuscript, we tried to validate the poly A tail length estimation from Dorado by comparing it to length that we know how long it is. But I think Dorado or Oxford Nanopore already provided their, or will provide their, I think yesterday someone asked it, will provide their training data on how they could validate the poly A tail length estimation, but also in our case we saw that the correlation was relatively high of the expected and the estimated length.
Yeah.
So we think that it's quite accurate.
Great. Did you guys want to add anything to that? Okay. And if somebody is looking to transition from cDNA sequencing to doing direct RNA sequencing, what would your advice be? Or is there anything, tips and tricks that they should be aware of?
We should be aware that there's a RNA flow cell for sure. Yeah. So in terms of poly A tailing, I think the direct RNA has an advantage because you have this ligation at the end of the. So when you do first-run synthesis, you do an annealing with the poly-DT adapter, and then you will have a ligation to the end of the read, and this makes sure that you are actually capturing the full poly A tail. And in cDNA, and like with the protocols, I know you have an annealing of oligo-DTs that are somewhere on the poly A tail, and then you do the first strand synthesis. So I think the direct RNA sequencing will be more accurate in terms of poly A tailing. But otherwise, there are high similarities between the cDNA protocol and the direct RNA protocol. So, yeah, I don't see big differences. So if you're trained in wet lab experiments,
So it's pretty
fine.
easy to
Yeah.
transition from
Yeah,
one to the other.
definitely.
Got it. Is there anything else you guys wanted to add to that? Okay. And I think now that we're doing so much epitranscriptomics and have all of these different RNA modifications, are there any challenges or common challenges that you guys come across in analyzing those? Johannes, I
Yes.
see you nodding your head.
So for the modified base calling, we did observe some false positives. And I think it's one main issue, if there are new RNA modifications introduced, to somehow ensure or validate with other methods to exclude those false positives. So especially for the new modification models, there is benchmarking needed, I think. And also tissue-specific benchmarking in terms of clinical context when we look in blood or any other tissues. And there, I think it's quite important to get orthogonal methods like GlorySeq or ETAM-seq or IVT as a negative control to be sure to get reliable results. This is a very important point, I think.
Yeah, no, that's a great point.
And I think what it will be a challenge again is to define thresholds for at which frequency or at which probability which modification can be really defined as a modification.
Yeah.
And this will be a challenge for the new models again.
Any questions from the audience? You could raise your hand. All right. So now that we've had, of course, a day and a half of London Calling, is there anything that stood out to you so far about direct RNA or epitranscriptomics in general?
So, yeah, I was impressed by the new models. I'm really looking forward to try to find out if we can really detect all the RNA modifications side by side. And I also like the talk of Eva Novoa in the epitranscriptomics session, where she showed that it's possible to, based on our ribosomal RNA modification, to find tissue cells in comparison to healthy cells.
Yes.
So just divide them just by a PCA. And this was very impressive. And I think that...
Yeah, I think actually this... So since we are detecting modifications on the read itself, and we have the read IDs and so on, we can really nicely now associate the modifications with specific reads and specific isoforms. And this is really a cool thing, because we can do it in different tissues, but you can also try for example, in gene expression to see whether certain isoforms have a modification or not. So this is a big advantage.
Absolutely.
Yeah.
Great. I think we're probably near the end of our time. So thank you for all the questions, and thank you so much for joining this demo, and a special thank you to you guys for joining our panel as well. If you do have any further questions about performing epitranscriptomics, direct RNA sequencing, do speak with me or any of our colleagues at our product tables. And now we'll be heading back to the auditorium, where you will see me again for more plenary talks, and to find out the winner of yesterday's spotlight session. So see you there.